Hi everyone, welcome to Bioorganic Chemistry. In the previous lectures, we have seen what is bioorganic chemistry and how it is different or related to other branches of chemical and biological sciences. We also had an introduction to carbohydrates. We have studied what is a carbohydrate and how carbohydrates are classified based on their functional group and number of carbon atoms present. So today's learning objective is to understand the molecular structure and their representation and to identify structural relationship between different isomers of carbohydrate molecules and learn how to interconvert open chain form of carbohydrates and their cyclic structures. So we begin with monosaccharides. What are monosaccharides? Monosaccharides are nothing but the simplest sugar units or simple carbohydrates. We cannot break them further. They are important source of energy, structure, and they're building blocks for disaccharides, oligosaccharides, and polysaccharides, and even nucleic acid structures. So they can be classified, as I said, based on the functional group, as we studied in the previous lecture, aldoses and ketoses, if they have aldehyde functional group or ketone functional group, respectively. We can also classify them as trioses if they have 3 carbon, tetroses if they have 4 carbon, pentoses if they have 5 carbon. Some of the important molecules, ribose and deoxyribose, they are present in deoxyribose is present in DNA, ribose is present in RNA. And hexoses, 6 carbon containing sugars such as glucose, fructose, and galactose, they are important source of energy. Now, very often when you look at textbooks or some research article or monographs, you see this kind of open chain structure, and this is called Fischer projection. You also notice sometimes this Haworth structure, the cyclic structure, or the same molecule. The example here is a glucose. And this cyclic structure in the cyclohexane form, a chair form of cyclohexane like. Then you might wonder what is the correct representation of this and how these different structures are related, how we can interconvert them. If you look at this structure, this particular structure is scientifically maybe not accurate because the bond angle here is about 90 degrees. But carbon is tetrahedral and then the bond angle is around 109 degree. However, this is a good representation of a 3-dimensional molecule in a 2-dimensional plane so that it's easy to write and understand some of the chemical reactions. So as we move further, we will learn how to interconvert these two using some simple methods. And I would like to stress that there is no need to memorize anything in this. We will learn as we move on and just by doing some simple transformation. So let us begin with this example here. And what we have here is a D sugar. And as you know, Emil Fischer has identified or discovered D-glyceraldehyde, which has the hydroxyl group on the right side. The definition is that when the hydroxyl group at the bottommost asymmetric carbon, this is the bottommost asymmetric carbon because this particular carbon is attached to 4 different groups or atoms. But this is the bottommost carbon, but this is not asymmetric. And this hydroxyl group at the bottommost asymmetric carbon is towards the right is the D. When the hydroxyl group is towards the left, then it is L. He also noted that in nature there are only D sugars, but there are no L sugars. So all sugars present in the nature are D. So now we can also think about the number of possible isomers when you have a one asymmetric center. And this is typically about if you have, say, n number of chiral center or asymmetric carbon, then the possible number of isomers are 2 raised to n because every carbon center can give you two possible isomers. And that is, in this case, we have one chiral center. So we expect two different isomers, that is D-glyceraldehyde, and the other form, which is L form. So we can write it here, which is CH2, OH, and OH. But this is L form of glyceraldehyde, but this does not exist in nature. That means in the nature, we can divide by 2, only one possible. Now we can calculate if we have a 2 chiral center for the next. This is 3 carbon containing, so it's a triose. Now if you, and it is aldehyde functional group because aldehyde is here, so it is a aldo triose. Now what if we have a tetrose? If we want to hypothetically write the structure of an aldo tetrose, how many isomers are possible that are present in the nature or natural, or in other way, the D isomers? Now, we have to remember the very definition of carbohydrates, and it states that the functional group is always at the top or close to the top, and the hydroxyl group at the bottommost chiral center is always towards the right. That means if you want to introduce a new chiral center, it has to be between the functional group and this chiral carbon. So we will have two chiral centers. 2. So that means we expect 4 isomers, but 2 of them will be D, 2 of them will be L. Since we don't have L form in nature, so we expect 2 D isomers. So we can start writing theoretically. We don't have to remember what is the name of that. Okay. And now we can look at the same structure. So we write CH2OH and OH here. And one possible isomer is this one. Okay. Remember, the hydroxyl group at the bottommost chiral center is always right side. So we can write the other form, which is, again, we have to fulfill the definition of carbohydrate D sugar, that is hydroxyl groups, always right. So the only change in the position or the stereochemistry of this hydroxyl group, and resting same, the functional group is at the topmost. So now we can calculate what happens if we increase one more carbon, that is carbon comes here. So now the idea is that we have one more, that 2 raised to 3. That means we will have 8, but in the case we have only half of them, they divide 4 possible D sugars. So we can write 2 more from this, 2 more from this. So if you continue to write, again, each of these will give you 2. So this is already tetrose, 4 carbon containing. We'll have 5 carbon containing and 6 carbon containing. So we can write. So we can continue to write this formula and see how we can construct this tree of carbohydrate. It's purely an hypothetical. So it's very easy to construct and write, say, for example, the tetroses. So here is what we have is D-erythrose. No need to remember the name. So what we have done is we have introduced one chiral center here, as I have shown you in the drawing before. So we have aldotetroses. Now each of these will give you, so remember, only the newly added chiral center hydroxyl group position has changed. Now we can continue further 2 from this. So that will give you this. Once again, everything, this backbone remains the same, and only the hydroxyl group position is different. This is the pentose, and when we go to hexose, each of these, so because here now the hydroxyl group is same side, all hydroxyl group are the same side. And the same thing is here, it's simply adding one carbon, just changing the stereochemistry at this stage. And so in total, what we have as we move on, in hexoses, if we go, there are 8 possible D sugars, 8 possible D sugar. They not necessarily all of them are present in the nature. They may present in small quantity in some organism also. So we can look how now how some of these structures are related. If I take these two, D-erythrose and threose, they have the same molecular formula. So if you write the molecular formula, they are same. They have different structural formula. That means the stereochemistry of this particular hydroxyl group or this asymmetric carbon attached to that asymmetric carbon is different. So we can now devise some relationship between these two. Because they have hydroxyl group and all the molecular formula same, slightly different structural formula, so therefore they are isomers. But they also have some specific isomer because the stereochemistry of one of the hydroxyl group remains the same, whereas only this center is changing. So therefore, we can say they are diastereomers. So now we can also write the mirror image of this. So D and L are always have an enantiomeric relationship, whereas this D and L trios have an enantiomeric relationship. Now we can also write the relationship between this and this carbohydrate molecules. So these are now Tetroses. So we can see an enantiomeric relationship and diastereomeric relationship. So now if you look at different aldoses, it's just an additional information. Allose, D-allose, which is a glycoside, generally is a conjugate, a sugar conjugate that is present in certain leaves or plant species. D-altrose, where one of the hydroxyl group has this position which is different from allose, is an unnatural monosaccharide. Rather surprisingly, this has been isolated from some bacterial strain. Glucose is the most important. It is produced in plants during photosynthesis and used to make cellulose in cell walls, which is the most abundant carbohydrate. And glucose is most important energy source for us. Mannose, also an important hexose or monosaccharide which is particularly important in addition to metabolism, also in certain proteins or glycoproteins. Another very rare hexose is gulose, which is found in some archaea or bacteria, but not abundantly. Idose is an exception, not found in the nature, but it's if you oxidize this, you get iduronic acid, and it is an important component in dermal sulfate and heparan sulfate, which are, as you know, heparan sulfate is important in many, including blood clotting, which are glycosaminoglycans. Galactose is a major component in dairy products, and another important aspect of galactose is that it is as sweet as glucose, but only 65% sweet as sucrose. Talose is another unnatural carbohydrate. Now we have seen monosaccharides. We have still not gone into the cyclic structure. We will come to that later. But the next group of monosaccharides are ketoses. Now, what are ketoses? Ketoses are those polyhydroxy ketones. So they have a ketone functional group. As we have seen, the functional group has to be at the topmost or close to the top. In the ketose case, it's always close to the top. And you also come across the cyclic structure represented like this, or you might be familiar with this 5-member structure, which you will see also in the case of nucleic acids. But now it is an interesting, what is the simplest triose or keto triose we can see. We have seen previously in the case of glyceraldehyde, right? So we have glyceraldehyde, which is an aldehyde functional group, which has the hydroxy group here, and which has already the building block has one chiral center. Now, the simplest keto triose or the building block is dihydroxyacetone. So we can write, first we can write acetone, which is CH3 and CH3. Once again, it's not the very correct way of representing an acetone, but it's easy to write and understand. So this as the carbon functional group here. Now it is a bit tricky because we have to remember how we can extend this structure. This has no chiral center. So then we have to remember what will be the next. That means what is our tetrose structure will look like. The definition says that in carbohydrates, the bottommost asymmetric carbon, the hydroxyl group should be right side in the bottommost chiral carbon. So that means we have, if you want to increase one carbon, that must come here. That fulfills the definition because the ketone group is close to the top or at the top of the chain. So what will be our tetrose if we want to write? So now we have a D. I'm talking about D. And it's a tetrose because we have 4 carbon atom and it fulfills, it is a D tetrose, D sugar. Now we can continue, but one important thing is when we had tetrose in the case of aldoses, we already had 2 molecules here, 2 D sugars. Because in the beginning, at triose case also we had a chiral center. Here we are first time now introducing chiral center. That means that we have half the number of possible D sugars in a case of ketoses compared to aldoses. Now, if you are increasing one more carbon, so we are writing pentoses. Now, the next carbon center should come here. That fulfills the definition of D sugar because this hydroxyl will remain the same always in the right side and the newly formed center. And now we have 2 chiral center, 2 raised 2, that means 4 possible isomers, but we can only have D sugar, so therefore we will have 2 isomers. How the 2 isomers will look like? So we write the original backbone structure same, and then one hydroxyl group here. And then we have ketone, then we have CH2OH. Then in this case, we can write one more structure. Again, the backbone remains the same, except the position of the hydroxyl group here will be different. So now we have written pentoses. The same way now each of them we can. Yes, so we have seen how to write and extend this tree, so we can look at it here. As I said, the new chiral carbon comes here, and the next pentoses, ketopentoses, 2 of them, and then we have hexoses. Here, some of the important carbohydrates are the sorbose and fructose, which is, of course, known as fruit sugar. So one point we have to remember, ketoses have one asymmetric center less than aldoses with the same number of carbon atom. That is because our initial triose already has no carbon center. In aldoses, it has a chiral center. So some key points about these carbohydrates or ketoses. This is a tetrose in this case. This erythrulose, xylulose is also, it is a pentose and then we have a hexose. This is a pale liquid which is naturally occurring in raspberries. It is also used in some self-sustaining cosmetics. Interestingly, xylulose present both as D and L enantiomeric form. L accumulates in the urine in patients with pentosuria due to deficiency in L-xylulose reductase. So this is a reducing sugar, just like D-glucose. We will come to that. So sometimes pentosuria patients have been wrongly diagnosed in the past to be diabetic because if the diabetic patients are also having a lot of glucose in the urine, so it can be mistaken for various reasons, because if you're using some test that detects based on the reducing power of the sugars that's commonly used, we will discuss about that, how this glucose sensing or detection is done. We also have a laboratory work about it. So another interesting sugar that I've chosen to tell here is Cyclose. It is a low-calorie carbohydrate, so it can be as sweet as that of sucrose, but it is a low calorie. So it is used in food and beverage. So it has 70% of the sweetness of sucrose. Fructose, as every one of us know, this is a fruit sugar found in all fruits and some, along with some other monosaccharides such as glucose and galactose, and they are directly absorbed to the blood. Then we have Sorbose present in fruits of certain plant species. It is also an intermediate for vitamin C synthesis. Tagatose is found in very small quantities in a variety of food. But interesting fact is that it has a similar texture and appearance to table sugar and is 90% as sweet, but with only 38% of the calories. So the ketoses are very interesting. We have seen in the previous example of Cyclose, and now we see here Tagatose. They're as sweet as the routinely used table sugar, but the calories are extremely low. We can have this group discussion a bit later, but we will continue now. So, in the beginning, I explained about monosaccharides, the open chain form, the Fischer form, which is not the very accurate way of writing here, but this open chain form exists. But what is important, this kind of open chain structure is existing only in about 0.02%. But then how this molecule exists in nature? They exist in cyclic form. And what are those cyclic form? And this is one of those cyclic form here. This is known as Haworth projection. And as you can see, and in this particular case here, I represented with a wavy arrow. It means this is a new chiral center, but the stereochemistry of this is not known, or it's a mixture of two isomers. So the question is always how we can interconvert if I given a carbohydrate like this to this, how easily. So without needing to memorize everything like this has to be like this. But if you are given any kind of this open chain structure of any sugar, can we write a cyclic structure? This is what we are going to see. So here is a very simple method for this, which I like to show you, is first to write a size structure, we can take some small steps. The first one is we can rotate this by 90 degree. And this 90 degree has to be in clockwise. So we can rotate and write this, the same structure like this. And then we write this hydroxyl group. So here, So this is carbon number 1, carbon number 1. When we rotate, this hydroxyl group carbon number 2 will be at the bottom. The hydroxyl group here, carbon number 3 will be at the top. The hydroxyl group here will be at the bottom. The hydroxyl group here will be at the this hydroxyl group will also be at the bottom. So we have H, H, and then we have this CH2OH group, which is CH2OH group here. So we have carbon 1, 2, 3, 4, 5. Now, if what we do, we want to get into a cyclic structure, so we can actually slightly rotate this to make into a ring-like or a 6-membered ring-like structure. So we can write like this. And now once again, we begin with the carbon number 1. Here is carbon number 1. We have carbon number 2 here, and the hydroxyl group is up. Here, hydroxyl group at the top, and hydrogen is at the bottom. Here, hydroxyl group at the bottom, hydrogen is at the top. And now we have this hydroxyl group at the bottom. We have hydrogen, and then we have CH2OH. So 1, 2, 3, 4, 5, and 6 carbon. Now what we do is something, because what we saw, there is a ring structure, there is an oxygen bridge. We will discuss what it is, but I'm simplifying. Now we rotate this particular carbon in such a way that this hydroxyl group comes very close to this, this carbonyl carbon, which is an aldehyde. So we can reposition this so that the hydroxyl group at carbon number 5 looks like this, and then we write it again. It is the same. So OH, H, this hydroxyl is up, this hydroxyl is down, this hydroxyl is down. Then this we have repositioned this for our convenience. And then there is the CH2. I can write CH2, or I can simply write an arrow. Now we have to remember, I discussed about when you have a carbonyl compound, I put R1, R2, a ketone or aldehyde, whatever. Now, if I treat with an alcohol or OH, it can attack from this side, or the alcohol can also attack from this side. The resulting will be either an hemiacetal or acetal. That means the both groups are functionalized. Now we have an aldehyde here, and there is an hydroxyl group, which is an alcohol group. So we can nucleophilically attack. And as a result, what happens? It is an addition reaction. So if you write that addition reaction, so we start to close the ring. During nucleophilic addition, we expect this, and there will be a proton that will go here. So we have this, and we have a new chiral center created because it was sp2, now sp3. The other thing here is an hydrogen. So we don't know, because the alcohol can attack from both sides. This carbonyl carbon here is sp2 hybridized. So it is a planar, therefore the nucleophilic attack can happen both sides. However, this can be, we can get one of the isomers predominantly based on the stability of the form structure, or also based on the steric repulsion that you get. So we write, now I write this CH2 as like this. This represents CH2, 2 hydrogens here, and OH. So we have now here the structure. cyclic structures. So very simple. The first is 90-degree rotation, any structure, then you exactly copy the same, and then you make slightly a cyclic structure. Now you might have many questions. Because it's a polyhydroxy compound, why only this particular hydroxy group attaching? Why not this hydroxyl group? Why not this hydroxyl group? Why not this hydroxyl group? So this is something I leave it for you to discuss at the end of this, and we will discuss in the next lecture in the classroom. Now we can look at it more carefully, what actually happens in this case. So we rotate it 90 degree, I said, this 90-degree rotation here, and it gives the same structure. And then you can write slightly the hydroxyl group, exactly what I was drawing in just before. And then we have to reposition this OH group, so that this is very closely aligned with the carbonyl group. So something like this. Now we expect a nucleophilic attack because of the lone pair of electron, and this is rather more reactive. And then we get a nucleophilic addition, we get an acetal. Now this is the carbon number 1. Now it creates a new chiral center, and that means at this stage we could be able to get more 2 isomers. And this particular carbon center is also called an anomeric center, and this process is result into isomers. This is exactly the same I wanted to explain you, that what happens when you have this kind of carbonyl compound. Because it is sp2 hybridized, a nucleophilic attack can come from this side or from the other side. So therefore, we will end up this way. That means 2 isomers. So we can look at what are those 2 isomers, and how which one of them are mostly present. So as I said that this open chain form is present only about 0.02%, but there are 2 cyclic form exist. The reason we already discussed, because of the sp2 hybridization of this carbon center, where the carbon nucleophilic attack can happen from both sides. So this is still open chain, right? So it is present in 0.02%. Then if you write the Haworth structure, you will end up having these 2 forms. The 2 forms, the hydroxyl group can be at the bottom, hydroxyl group can be at the top. And this form is called alpha, this form is called beta. So now we see the beta form is more stable compared to the alpha. It exists in excess. If you dissolve glucose, that can already be explained by many ways. Here, if you look closely, they both have very close, and there is a steric repulsion between them that might explain they're not that stable, that the stability, and when it is beta, we will have only hydrogen here. So therefore, the repulsion is much slower. If you make a space filling model of this in 3D, you will really see this kind of interaction. So now, what is this structure? This is a pyranose form coming from this particular 6-membered ring structure here. And because it is glucose in its pyranose form, so we call them as glucopyranose. Now, this is alpha D-glucose in pyranose form, so we call alpha D-glucopyranose. It's in beta glucose form, so this is beta D-glucopyranose. So now we have seen that how to get this structure. There is one more structure, that is the chair form of cyclohexane-like structure. So how to get that structure? So essentially you write a cyclohexane-like structure, very simple, very same as writing a cyclohexane. Now we can, this is the same oxygen. Here is carbon number 1, carbon number 2, 3, 4, and 5. Now we know that this hydroxyl group at the bottom, so we write at the bottom. This hydroxyl group also bottom, we write like this. This hydroxyl group at up, so we write at the up. This hydroxyl group at the bottom. right? And this is CH2OH is also up. So what else is here is hydrogen, 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 and same. So now this is alpha, so it's also alpha. Same we can write beta, only the position of this is different. Now if we take cyclohexane, okay, we will also look into the model of this cyclohexane ring. There are two things that we have to remember. If I draw a line like this and like this, we will always have two types of orientation. One is called equatorial. Equatorial. That is everything aligned along this line. And the one is axial. Axial is the one which is going vertically in this. So this hydrogen which is up is in this case is axial. This hydrogen is also axial, but this is equatorial, this is equatorial. This is again equatorial, this is axial. So if you look at this hydroxyl group, all of them are in equatorial position. And that is when you go to beta, even in this alpha case, this is in axial because it is in this, but in beta case, this is in equatorial position. So that also makes sense of the stability of this. We will see soon about this as well. So now here are the structures. As I said, the hydroxyl group, this is the alpha glucopyranoside, this is the beta glucopyranoside. The hydroxyl group is at the equatorial position. Now, many of you have seen this 5-member-like structure. So if I take instead of a 6-carbon containing, simply we take a 5-carbon containing sugar. And this is a ribose. Ribose is an important component in ribonucleic acid. So you see almost all textbooks with ribose and sugar in it is represented like this, where there are 5 carbons and the hydroxyl groups are oriented like this. And this kind of structure, because it's coming from furan, it's called furanose. So if I take a ribose, we can do the same thing. We can simply rotate it by 90 degrees and then see how we can construct structures. And we have carbon 1, 2, 3, 4, 5 is the same thing as we have here. And now we make a 5-member ring-like structure. And then once again, the nucleophilic attack will happen here. And this will lead to the 2 isomers coming. So once you have familiarized how we write 6-membered, and this is the tetrahydrofuran ring, that's where the furanose comes. The ribose structure. So once again, we get 2 types of isomers, 5-member alpha and beta. Now this is a ribose sugar, and this is an alpha D-ribose because hydroxyl group is down, and it's a furanose form. So it is alpha D-furanose. This is a beta because hydroxyl group is at the top. So it's a beta D-ribofuranose. So the idea is that you take the orientation of hydroxyl group, alpha or beta, D sugar, most of the natural sugars, and whether it's ribose, glucose, fructose, and then the type of the ring structure. So now we can also write the same way for fructose, right? If we are able to do that, we can also write fructose. How can we write fructose structure? This is a good exercise for you. Can we use the same way? Yes. So now fructose has an interesting structure. It can also form 5-membered ring. That's because if we take the functional group, in this case is carbon number 2, unlike glucose where the functional group was in carbon number 1. So now if you think that the hydroxyl group from 5, which is here, has to attack, then we expect to form a 5-membered ring. But it is also possible that this hydroxyl group, this is why I asked, can attack and form a 6-membered ring. Now, already you can imagine that when you have this, we expect 2 isomers, alpha and beta. Here also we expect 2 isomers. That means there will be multiple forms of sugar will coexist when you dissolve in a solution of a fructose. So fructose can exist as Furanose form. Remember this Furanose alpha and beta. So the alpha beta is based on the hydroxyl group position, not this functional group. And then we can also get Furanose form, alpha and beta Furanose. Open chain, alpha, beta, Furanose, and Furanoses. So now I want to leave you with this question. Hexoses can form 6 and 5-membered ring. Which of them are relatively stable? That is the first question. So which of them are stable and why? You can say any of them, but you have to justify your answer. Then fructose forms 5 and 6-membered ring. That means why not glucose form 7-membered ring? Because in fructose we saw the primary hydroxyl can attack and form a 6-membered ring. Is it possible? If yes, why? Or it is not possible? If not, why? Please come up with your answers and see you in the next class.